Hello everyone, my name is Emily. I'm another member of the outreach team, and today I'm going to tell you the things we need before we head out to the tide pools. So first, we have a quadrat, and this is going to define your research area out on the tide pools. Here is our clipboard, and this will have our Intertidal field survey, as well as some other materials that you're going to need. And another thing you need to make sure you have is some sort of boots or shoes you don't mind getting wet, because we'll be walking along slippery rocks, and there's a good chance your feet will get wet. Lastly, we have our tide pooling kit, which has a ton of different supplies that you'll need out there that we'll talk about later. Alright, so now that we're out here at the tide pools, we're going to measure the temperature of the water. So I'll leave this here for a couple seconds. Okay, the water is 10 degrees Celsius, so we're going to write that down. So next we're going to measure the salinity of the water using a hydrometer. So to do this, you have to hold it flat to make sure it gets filled. Okay. So you'll see the needle moving around a little bit. Sometimes you have to tap it. Okay, so it looks like the salinity is around 32 parts per thousand.
After all the abiotic measurements have been taken, salinity, temperature, pH if you're doing that, then you're going to move on to biotic. So we're going to use this quadrat, this square of PVC, and that's going to be your entire study area that you care about. Any of the seaweed and mollusks that are outside of that, you don't have to pay attention to those at all. For your data sheet, you just want to record what is inside your quadrat. And there's a lot of different types of seaweed and critters in here, so you're going to use the ID guide that we give you in the tide pooling kit and start with seaweed. All right, so the first side of your data sheet has seaweeds and algae. So start by identifying one of them. I'll start here. This is rockweed. And what we're going to do with the seaweed, instead of trying to count the individual fronds of it, or strands of it, we are going to do a percent cover. So we're going to look at the rockweed and say, I would say that it's about 10% of this total quadrat is covered in rockweed. Okay, so then we're going to write 10%. I'm in the low intertidal right now, and so we are going to be writing this in the column that says low intertidal. So we're going to write 10% rockweed. I could look for one other type. I see just a very, very tiny bit of sea lettuce on here, and that's maybe 1% cover of sea lettuce. And then you would continue that for all of the types of seaweed that are in your quadrat, again, using the ID guide. Once you've done percent cover of all of the seaweeds, then you're going to move on to the critters. So you're going to identify, do one species at a time. So in this case, I'm going to start with this little guy right here. And if I look on my ID guide, that is a smooth periwinkle. So he's pretty defined in his color. They're usually yellow, can be a reddish, orange, almost a green sometimes. And they have a nice smooth shell. So I'm gonna look around and see if I see any other snails that are like that in the quadrat. Sometimes you wanna move aside the seaweed a little bit. I actually don't see any other smooth periwinkles, do you?
I do not.
I don't think so. All right, so we're just gonna write down one. Then I'm gonna move on to this other type of periwinkle here. These are our common periwinkle. They are usually gray or brown in color. They have a more defined point. And then I'm gonna get started counting. Luckily, you'll be doing this in groups, so two people can be working on counting different things, because as you can see, there's just a huge quantity of these common periwinkles in here. If I were to count them, you know, I could estimate, I don't know, maybe 35 or something like that. Then you'd move on to, here's some blue mussels. This mussel actually has a barnacle on it. barnacle's a little open right now. See, it's filter feeding. So you just move through the sheet, identify everything that you can, make sure to move rocks out of the way, move the seaweed out of the way. Sometimes you'll find crabs and other things underneath the rocks. But just make sure you categorize and list out everything that exists in your quadrat. After you've done that, then you can take a little bit of time to explore outside the quadrat and see what else you can find.